it's hosted by Ed Wild. It was originally conceived in June as a presentation that the 5 HDBuzz editorial members did, but it's just me today, so it's a solo news presentation. So I'm the anchor, and I'm going to tell you what's happened, and that's the first segment. The next segment is what's next, and then we do have an outside broadcast segment from Rachel Harding, but that's actually, I've just realized that's not going to work, but let me have a think about that. We might just have to skip that, and I'll tell you what Rachel's talking about. So here are the headlines. Highs and lows for Huntington lowering trials. Results of the Proof HD trial of predopidine. Valbenazine emerges as a new career treatment. A new generation tominersen trial underway. CAGs in the crosshairs. Stopping Huntington with a pill. Okay, the first segment is what's happened. So the first thing I'm going to talk about is the highs and lows of Huntington lowering trials. And there's been a lot more lows than we would have liked. But we are, I think, emerging from the lows into a new era where we can start to expect some good news. And the basic principle of all of these Huntington lowering trials is that Huntington is the name of the protein that causes Huntington's disease. And each person who has an HD expansion will generally have one healthy copy of the gene and one expanded copy of the gene, which produces this protein called mutant Huntington. And the idea of Huntington lowering is that we want to stop the brain from producing as much mutant Huntington, and therefore ideally delay or slow down the progression of Huntington's disease. And when I first started seeing patients in the HD clinic in London in 2005, my patients had just started hearing about this kind of thing and were asking me how long it would take before this became a reality. And I very optimistically said, 5 to 10 years if we're lucky. And then the first dose of the Huntington lowering drug, tominersen, which is the one that most people have heard most about, was given 9 years and 11 months after I started. So these things take a long time. That was 18 years ago now. So we've been in the era of Huntington lowering for 8 years. And there was a lot of excitement. And then, as I say, we went on a bit of a roller coaster ride. But the basic principle is that each cell in our body contains DNA, and that DNA is a recipe for the mutant Huntington protein. But what we're targeting is this structure in the middle called RNA, and that's kind of message molecule or a working copy of the gene. And the drugs that lower Huntington basically interfere with that RNA. They either stick to it directly, or they monkey around with the machinery that handles that RNA. And one way or another, if you get rid of the RNA, less of the protein is made. And the three basic categories of Huntington lowering drug are spinally delivered ASOs, which I'll come back to, viral gene therapy, and small molecules. All molecules are small, but small molecules are smaller than big molecules. So what is an ASO? An ASO is an antisense oligonucleotide, and that's why we call them ASOs. And it's basically a strand of chemically modified synthetic DNA, which has been designed to stick to the message molecule that comes from the Huntington gene. And when that happens, it tells the cell to delete the RNA and make less of the protein. And back in 2017, the most famous bit of news about ASOs was that they work in terms of they are actually able to lower the concentration of Huntington in the brain. Unfortunately, the more recent bit of famous news about the ASOs in HD has been that there have been several quite high-profile negative results. Most prominently, 3.5 years ago now, believe it or not, Roche pulled the plug very, very abruptly on the tominersen trial. That drug had been called Ionis HTTRX, and then it's now called tominersen. The reason for the early termination of that trial was that the patients on frequent dosing of the ASO injected into the spine were actually doing worse than the patients who were receiving placebo injections. What we want here is, so the gray dots show the gradual worsening in patients who were on placebo, and we would like the blue dot to be above the gray, and we'd like the orange dot to be above the blue. And if that had happened, it would have meant that the drug was working. In fact, if you turn this graph upside down, the trial was a huge success. I can joke about it now, but it was, heartbreaking at the time. But the only failed trial is a trial that you don't learn as much from as possible. And not only the people like me who helped to run the trial, but particularly the patients and their families gave a huge amount to enable this trial to occur. And it really is an ethical duty to find out as much as possible about what the trial actually told us. So we know that drug at that dose in that group of patients didn't work to slow the progression of the disease. But one of the decisions the company then had to make was, do we abandon this drug altogether, or are we going to try and rerun the trial in some different way that would be reasonably expected to produce benefit? And once the dust had settled and the data had been thoroughly combed through, it became apparent that the major issue was that probably people were given a bit too much drug too soon. And in the context of a vulnerable HD brain, that ended up doing overall more harm than good. And obviously what we want is a drug that does more good than harm. So if you look at the graph on the left here, you can see this is what I just showed you. If you look at everyone, all 800 people in the trial all at once, overall, it didn't do any good. But the company Roche then basically decided to split the group of patients into 4 roughly equal groups, dividing them first on the basis of age, young patients versus older patients, and then on the basis of CAG repeat, essentially. So it was, there were 4 groups: young people with low CAG, old people with low CAG, young people with big CAG, and older people with big CAG. And if you just focus, you would expect that the people who were young, who also had low CAG counts, might have the healthiest brains relative to the others, and therefore might have the ability to experience some of the benefits of the drug without experiencing so much of the damage that was apparently caused by the high doses. And much to everyone's surprise and pleasure, that was what was actually found. Now, this is what's called a post hoc analysis, which means it occurs at the end of the trial, after the trial has concluded, and therefore it can't be used because it wasn't pre-declared as a potential endpoint. This can't be used to make any decisions about licensing or any declaration that the drug works. All it is essentially the company sharing its own thinking about whether to ditch the drug or give it another go. And what was found was, as you can see on the left here, gray is at the top, orange is in the middle, and blue is at the bottom, and that's not what we want. The 4 monthly patients were the ones who received the drug less frequently. 2 monthly received it more frequently. And so if you just look at the week 69, which is a year and a bit, results here, the gray patients, again, were a little bit worse at the end than they were at the beginning. The blue patients, which are the 2 monthly, the frequent dosing, were again worse than the placebo patients. And that was even if you only look at the young people with low CAG. The orange patients were the less frequently dosed patients. And actually they ended up doing, on average, their scores were basically stable over the course of the trial. This is the clinical rating score. And the orange is basically just about on the side that we would like of the gray. What that means is that there is this is a kind of suggestion that theory that we developed, which is that people who are younger with lower CAG counts might be more resilient in terms of the ability of the brain to experience benefits of the drug, potentially would be borne out. This, like I say, this isn't a result in itself, but it is a finding that allows the design of another trial. And I'll come on to that later in the future-looking segment. What are we looking at here? This is another result from the post hoc analysis, and this is looking at the level in blood of a protein called neurofilament light. Now, neurofilament is a measure of damage to brain cells. So during the progression of Huntington's disease, neurofilament is released into the spinal fluid by the brain because neurons get damaged, and then it finds its way into blood, and you can actually measure the level of neurofilament in blood. And that's what we're looking at here in the Generation HD1 trial. So what happened, and everything here has been adjusted to look at the level of neurofilament relative to the patients who were on placebo. So the dotted line here represents what happened to the placebo patients. And basically with neurofilament, the key thing you need to know is up is bad, because that means neurons are damaged. Down is good, because that means that neurons are being preserved or are dying less frequently. And if you just look at the blue blobs, which is the 8-week group, so that's the more frequently treated patients, you can see that the neurofilament had a big increase and then went back down and potentially was heading below baseline at the point where the trial was terminated. But we don't know because the trial was terminated. The orange is interesting, though, because there's again a smaller increase in the less frequently treated patients. So something bad is happening, but then it more rapidly falls to baseline. And actually, if you look at week 101, the dosing was stopped at week 69, but at week 101, the neurofilament level in blood is actually below baseline, which, again, this is a post hoc analysis of a trial that was terminated early, so we can't conclude anything from it. But if you want your neurofilament to be anywhere at the end of a trial, it is certainly below baseline, because that is a suggestion of potential benefit. And that's in this whole 4-monthly treated group. Again, it doesn't prove that the drug works, but I think it is a suggestion that there is merit in doing another trial of this, the first Huntington lowering drug to work. So around about the same time as the Roche program kind of went off the rails, there was another bit of disappointing news, which was that the WAVE ASO trial had also been terminated. Now, you'll recall that the Roche tominersen program was stopped because although the drug was lowering Huntington, that was not producing benefit. The WAVE trials were stopped for a different reason, similar drug, but different reason. And that reason was that the drug simply was not lowering the Huntington level at the highest tolerated dose. And when that happens, there's basically no point carrying on, because if a Huntington lowering drug doesn't lower Huntington, then it's not really a Huntington lowering drug. It's just a molecule. And so the company stepped back and then basically decided to go back to the drawing board and redesign its ASO drugs, but with a new chemical formulation, which I will return to in due course. But this is a recap of what's happened so far. Now, earlier at the beginning of this year, there was another bit of disappointment. Now, these ASOs have to be injected into the spine, which is fine if that's the best we can do. But if we could accomplish something similar with a pill, then clearly that would be a much more desirable thing to do. In fact, if I had, when I was giving the first injection of ASO in 2015, if someone had burst into the room and said, hang on, we've got a pill that can do the same thing, I probably wouldn't have carried on with the injection. Now, I think it's really important, given our experiences, that we try everything, and we do so in parallel so that we're not, waiting around for something to work or something else to fail. Novartis entered the Huntington lowering scene quite recently, but with a drug that they had developed for another disease called spinal muscular atrophy. And that drug was called branaplam. And along the way, they discovered essentially by accident that branaplam was capable of lowering the production of Huntington protein. And so, needless to say, there was a lot of enthusiasm for testing this drug in HD patients, and I think it was a good decision because it was important to find out whether this drug could lower Huntington in a way that might be meaningful. And the drug works in a slightly different way. It's called a splicing modulator. It basically changes the way that the RNA is edited. The message molecule is kind of snipped up and joined together, and it does so in a way that basically gets rid of the message molecule and causes the protein not to be made so much. Unfortunately, towards I think it was either the end this time last year or early 2023, the company had to pull the plug on that trial because the drug, it turned out, was causing damage to peripheral nerves. The peripheral nerves here are, as opposed to the neurons of the central nervous system. So we think about the central nervous system, which is the brain and spinal cord, and then the peripheral nerves are the ones that run it through the body and into the arms and legs and everywhere else. The extremities, you might say. Unfortunately, Novartis had to report that branaplam was, for some reason, causing harm to the peripheral nerves, and that was causing a big increase in neurofilament, NFL. And as you'll remember, neurofilament going up is bad. So although that was a bad result, what it did tell us was how sensitive neurofilament can be as a safety marker. So because in the tominersen program, and now again in the branaplam trial, neurofilament turned out to be a really useful canary in the coal mine. When neurofilament goes up in one of these trials, if you don't have a good explanation, it's really something you need to pay attention to. So branaplam no longer under investigation as a treatment for Huntington's disease, but could potentially tell us a lot about how these drugs work and how we might be able to make better drugs to lower Huntington in the future. Now, it's been, so those are the ASO drugs and the small molecule pill drugs. There's a sort of second tier of complexity and intensity when it comes to Huntington lowering, which is the idea of gene therapy. None of the things I've talked about so far are gene therapy, which technically is defined as introducing new DNA into the body of a person. And the leading company that's doing gene therapy in Huntington's disease, it happens to be a sponsor of this meeting, but I didn't know that when I put my talk together. But nonetheless, they are the only company doing gene therapy right now in human patients with HD. And believe it or not, they've been doing it for 4 years. Actually, it's 3.5, but anyway, it's a while. So we're in a new era. Why would we bother though? So gene therapy involves lengthy neurosurgical procedures, which you can see happening here on this MRI scan. A thin tube called a catheter is inserted deep into the brain, into the part called the striatum here, which is the part that's most affected in Huntington's disease. And then down the catheter is injected a virus. The virus contains instructions for making a Huntington lowering drug. So the neurons are turned into a factory that makes its own Huntington lowering drug. So the neuron becomes the cause of the problem and the solution to its own problem, in theory. Why would we bother with all of this complexity if we have a pill? Well, the answer is, if this works, it would be a one-off treatment that would last lifelong. No one would ever have to remember to take pills, and potentially it could be done many years before symptoms begin. And because it's working all day, every day, it could potentially be part of a future in which people have either not to worry about HD or have to worry a lot less about it because their brain is basically treating itself. So it's a bit of a moonshot, but you know, high risk, high reward, I would say. So where are we up to with uniQure? Well, it's been a busy year. Around about this time last year, the study, the uniQure reported that they'd had to pause the study because there'd been a few instances where there'd been more than the expected amount of inflammation in the brain after about a couple of weeks after these operations were done. So you can imagine, you go into hospital, you have a long 12-hour neurosurgical operation, you feel okay, you get sent home, and then a couple of weeks later, a few of these patients were having to come back into hospital because they were unwell with things like headaches or other symptoms. And then when the scans were done and the lumbar punctures were done to look at the spinal fluid, there was evidence that there was inflammation happening. And it's not a big surprise because, like I say, these treatments are injected into the brain, packaged in a virus. And, it's not a big surprise that you stick tubes in someone's brain and then you inject a virus down it. There's a bit of inflammation. So the question was, is this inflammation a deal breaker? Is it too much to deal with? Is it going to, potentially offset the expected or hoped for advantages of the treatment? Or is it something that we can manage? Can we reduce it? Can we handle it? Can we live with it? And so that was what was happening this time last year. Since then, the trial has been restarted with a tweaked protocol, which enables a short course of steroid treatment to be given around the time of surgery, which is an anti-inflammatory treatment. And the aim of that is to make the brain less inclined to overreact to the presence of this harmless virus. And then just a couple of months ago in June, uniQure announced the data from the first updated data from the first cohort of patients. So the trial's a bit complicated because it has to have a different design in Europe and in America. In America, patients have been randomized to either active treatment with the virus or to a sham surgery in which they just have little incisions and sometimes holes put in the skull, but that's it. I mean, that sounds like quite a lot, but it's a lot less than having the injection into the brain. In Europe, the European Medicines Agency didn't like the idea of drilling holes in people and not giving them a drug. So everyone in Europe gets either low dose or high dose treatment. So it's a slightly complicated two cohort thing. But this is what was presented. Now, before you look at the graph, I want you to look at the numbers at the bottom. These are the number of people that were studied at each time point. So even at the very highest, it's 6 in the low dose group, 10 in the high dose group, and 10 in the control sham treatment group. So those are tiny numbers of patients. If you look at the more recent time points, because some patients were treated later, the numbers are even smaller. So from the 15 and 18-month cohort, there's no patients who remained on control treatment in the control arm that long, but the numbers in the treatment arm are down to 6 and 2. And that's not because they've died or anything. It's because they haven't reached that far ahead yet. So, and there's only 5 patients who've actually reached the 2-year time point and no controls. So these are tiny numbers, and the absence of controls later on, because after a year, the control patients are then invited to roll over into into active treatment. The absence of controls later on makes it all rather difficult to interpret. So, but what you can see, this gray triangle here, it gives an estimate from previous studies of roughly how you would expect the patients to progress based on this overall UHDRS composite score. And as you can see, as before, down represents worsening, up represents improvement. And what we're seeing is in the low dose arm, which is purple, there is a suggestion that the patients here, if you look at the start of the trial, that we reset the numbers to 0, and then at the end, it's still roughly 0. They haven't declined much. And now, remember, only 5 patients after 2 years. So maybe these were patients who were going to do fine anyway. But the gray line is matched to patients who looked approximately the same in various natural history studies. So it's certainly good for the line to be here rather than below, but 5 patients is not enough to draw firm conclusions. Excuse me. And in the high dose group, we've got 6 patients at 18. I'm sorry, we've only got 2 patients here at 18 months, and we've got 4 patients here at 15 months, and we've got 9 patients at 12 months. But again, the blue line is above the gray where we want it, and it's also good for the blue line to be above 0. But these are not enough patients and not enough time to make conclusions. We need to step back from all of this and remind ourselves that this is the first ever gene therapy trial in Huntington's disease, and it involves half a day of neurosurgery and the injection of what is essentially an alien virus into the human brain. So a good result here is everyone didn't die. I don't know if anyone from Unicure is listening. I don't know whether they'd be happy if I with me saying that. But my point is, this is a high risk, high reward strategy, and it's possible that the Huntington's disease human brain could have reacted really badly to this whole situation. And the good news for me, the really, really, really good news is that we're now two or 3 years into this trial, and so far it seems to be pretty well tolerated. It's safe, and the one hiccup that we had, which was all this inflammation, we now have a good plan for managing it. It is a slow burning process. We cannot rush into making this drug approach fast or easier to deliver. But these, it's something that will slowly, hopefully build and build and build with time. And the great thing about this kind of gene therapy is that because it's a one-off treatment, patients basically, as long as they're alive, will remain in the trial forever. So, these patients will still be of interest 10 or 15 years from now. And so slowly, even if the numbers remain small, the passage of time should make the picture a bit clearer. This is looking at the neurofilament in the Unicure trial. And as you can see, it went up and then it came down. Why did it go up? Because the patients had brain surgery. And if you damage the brain with a catheter, even in a very controlled way, it's inevitable that some neurofilament will be released. So this is not a surprise. Unlike in, say, the branaplam trial, where it was expected that the neurofilament would be flat, it went up, and that was bad. Here, it's kind of expected, but also we don't want that neurofilament rise to be too big, and we want it to come down as quickly as possible. You can see in the high dose group, it went up a reasonable amount, and then it actually it's pretty slow to come down, so that it's only really by sort of 15-18 months that it's actually back to baseline. That is longer than we would like, and that's partly why the steroid treatment has been added, with the purpose of trying to bring the neurofilament down. And again, tiny numbers of patients, but after 24 months, the purple is below baseline, and that's where we want it. But we don't know if it will stay there, and we don't know what that means. News from the measurement of Huntington in this trial is a bit confusing, and it's not clear why, but the Huntington level seems to it seems to be going down and then perhaps less reliably down. But again, look at the numbers here; they're tiny, and the system used for measuring Huntington is a bit less than perfect, and also the volume of brain that's been treated is actually tiny compared to the volume of the whole brain. So I would say this is all rather difficult to understand. But the basic message for me is the trial is happening. Patients are tolerating it well, and the procedures are becoming gradually more and more finessed. Okay, so the next headline is the results of the Proof HD trial of pridopidine. Now, pridopidine has been in trials for as long as I've been in the Huntington's disease field, so at least 18 years. It's a drug that was initially thought to be changing the way that dopamine acts in the brain. Later on, it was discovered that it maybe has another mechanism acting on sigma-1 receptors, which has something to do with the health of neurons, and may be involved in Huntington's disease. And so it was kind of hoped that it might be something that could actually slow the progression of Huntington's disease as measured by the total functional capacity. And that's what the Proof HD trial was looking at. The trial read out earlier in the year, and unfortunately, it was a negative trial. The drug didn't meet its primary endpoints or its secondary endpoints. So we're again in the realm of trying to figure out whether there might be a signal of potential benefit, or is there potentially a benefit in a subgroup of patients. And what was presented in June was actually a slightly extended analysis. And on the left, you can, and this is a photograph of a presentation because I had to take a photo while it was being presented live. But patients on pridopidine are purple, and the TFC was not on the side of the blue placebo line that we would like. So that's a negative result even after extended follow-up for TFC in the overall group. But there was known from a previous trial, it was known that there was a potential interaction between neuroleptics, which are also drugs that act on dopamine. And these are drugs like olanzapine and risperidone. So these are drugs that are very widely used in HD. and we basically use them mostly for treating either chorea or treating problematic behavioral symptoms like irritability and aggression, and sometimes both. So they're widely used, and we generally use them when we have to. In other words, we're not kind of giving these drugs out idly. We use these drugs when it is important for improving quality of life. And sometimes these drugs are necessary for securing the safety of the patient or the people around them. So they're an important group of drugs, but they do have this apparent potential to interact with predopidine. And so if you look on the right-hand side of this graph, you can just look at the patients who were not taking predopidine, neuroleptics or similar drugs. And on this occasion, the line is where we would like it to be. It's above the blue line. However, even though these are large-ish numbers of patients, this is not what we call statistically significant. So unfortunately, we can't really conclude anything from this in terms of efficacy of the drug. The company which is Prolennia, remains committed to exploring whether this drug could have benefit potentially in this subgroup of patients. But we don't yet know whether they will need to run another trial or whether they will have the enthusiasm and resources to run another trial of predopidine. It remains to be seen. There was good news, though, from another trial of a drug called valbenazine. And now valbenazine, it might remind you of a drug called tetrabenazine, which is used quite a bit in the UK. Tetrabenazine is one of the earliest drugs for treating chorea, and it works. It's not a strong or particularly powerful drug, but for many people, it does make a difference to the amount of chorea. Tetrabenazine, most people have to take it 2 or 3 times a day. Valbenazine is a related molecule, but only has to be taken once a day. And the good news about the valbenazine trial is that not only did it produce a significant decrease in chorea scores compared to placebo, but the FDA in America has recently licensed valbenazine. So it's now the latest drug to be licensed for the treatment of chorea in Huntington's disease. It hasn't been compared directly with tetrabenazine. Hopefully that will come in time, or we can, generate enough data to figure out which patients the drug works for. But it's really good news. There's another drug, safe, well tolerated. It works for chorea, and you only have to take it once a day. I would expect that the company would be interested in licensing that drug in Europe, but I don't yet know whether they plan to or what the timeline is for that. Okay, so now it's time to look into the future. What's next? Science never sleeps. While I'm here in my living room in East London, there are people all around the world keeping stem cells alive and doing drug trials and all sorts of things. So what's happening now and into the future? So several Huntington lowering trials are underway. Happening now, dosing is happening. Let me give you an overview of some of the more prominent ones. So tominersen, the drug that was negative in the Generation HD1 trial, is now in a new trial, Generation HD2, exploring new doses. Vico Therapeutics is a newcomer, which I'll go into in detail, and PTC Therapeutics is actively running a trial of an oral Huntington lowering drug. So Generation HD2 is testing what the sponsor is describing as a refined hypothesis for tominersen in Huntington's disease. It takes the exploratory findings from Generation HD1, uses a more focused population, lower doses and less frequent doses, and is really focusing on the lessons that were learned in terms of safety and biomarkers from the previous trial. So the comparison is here. At the top in green is the dosing regime that was in the Generation HD1 trial. And you can see that the doses were 120 milligrams, and there were 2 doses close to each other at the beginning. In the Generation HD2 trial, the highest dose is 100. Also being tested is a 60 milligram dose, and all of the doses are 4 months apart. So it's a lower dose, but the company reckons that should be enough to hit what we call the therapeutic window. And because it's looking at somewhat younger patients with lower CAG repeats, it's hoped that we could avoid the early damage that seemed to be happening. Does this mean that people with bigger CAGs or people who are older will never be able to benefit from tominersen if it works? Absolutely not. What it means is that the company is simply focusing on the population where it is most likely that we'll be able to show a positive result if there is one to be found. When that happens, if that happens, it will then be a matter of priority to see how far we can push things. Can we go higher? Can we go older? Can we go bigger CAG? It's a journey, and it needs to be. We need to do this first trial to figure it out. CAGs in the crosshairs. So what's this about? CAGs are the cause of Huntington's disease. Too many CAGs in the gene. The gene is a recipe for the protein, but Huntington's is not the only is not the only disease that has CAGs, too many CAGs in a brain gene as the cause of it. There's actually numerous diseases, including things like spinocerebellar ataxia 1 and 3, which are also caused by CAG expansions in other genes. And this company, Dutch company Vico Therapeutics, had the idea of saying, well, why don't we produce an ASO that targets the result of these CAG repeats? Because if that works and if it's safe, it should work for all of these diseases. Again, this is a little bit of a high-risk, high-reward scenario, because one of the things that will inevitably happen if you use a drug of this kind is that you will end up interfering with genes that are functioning okay. So if you have HD and you take this CAG targeting ASO, hopefully it will lower the Huntington protein, but it will also lower the production of proteins which cause these other conditions. Is that a good idea or not? I think it's a good idea. The question is whether it's going to do more good than harm or more harm than good. At the moment, we can lower Huntington, but we haven't been able to produce benefit. So what we're hoping to achieve is lowering Huntington safely and doing so in a way which actually might target some of the more toxic versions of the Huntington protein, which happen right at the beginning of the gene, or rather which come from instructions at the beginning of the gene. And because it targets the CAGs, it should actually also be a little bit better at lowering the mutant proteins than the wild type proteins. So there are reasons to go ahead with this trial, and it's being tested in what's called a basket design, because these three conditions, spinocerebellar ataxias 1 and 3, and Huntington's disease, are being tested in parallel in the Vico Therapeutics trial. This is how many people are expected to be enrolled. It's an early stage trial. At the moment, it's focusing first on the spinocerebellar ataxias, but then Huntington's disease patients will start to be recruited into the higher dose arms. This trial will be happening in the UK. It's already running on mainland Europe. And hopefully it will be coming soon to a country near you. So what about this pill, PTC Therapeutics? The drug that they've been developing for ages is called PTC 518. And there's been a lot of excitement about it, but it's taken a long time to get into trials because the company is really particular about what kind of data they need before they can actually run a trial in humans. This trial is now finally underway, and it's called Pivot HD. And to cut a long story short, it is a pill which messes with the splicing of RNA in our cells, and in the process reduces production of the Huntington protein. The trial started enrolling roughly this time last year, and is aiming to recruit 162 people, each of whom will be dosed for just 3 months, but then they will be able to roll over into an open label extension to the trial. And it's looking for safety, but also early signals of effectiveness. And actually, in this trial, we had a really, really, really encouraging update. 2 pieces of really good news. First, for the first time in patients, a pill was successfully shown to be reducing levels of Huntington protein. This is measured in blood, but the drug also is known to get into the brain. And so we hope that we will soon be able to measure reductions in Huntington in the brain of these patients. And what they showed was that with a 5 milligram pill, there was about 20% reduction. With a 10 milligram pill, it was about a 30% reduction in Huntington, which is exactly what we want to see. So what about neurofilament light? Well, the company showed this slide in June, and there's a suggestion that the neurofilament is below baseline, which is where we want it, in both of the treated, both of the treated arms. However, I'm skeptical of this because the numbers are small. And if you look at the individual trajectories, there isn't a big difference. But basically, the neurofilaments more or less horizontal. What is really good news about this, though, is that because this is another Huntington lowering pill, which is also what branaplam was, and you'll recall that Novartis had to pull the plug on branaplam because it was increasing neurofilament because of damage to the peripheral nerves. This slide is really good news because what it's showing us is that after 12 weeks of treatment with high dose and low dose PTC 518, there is no increase in neurofilament. In other words, there is no sign so far that it is damaging peripheral or central nerves. And that for this drug is really good news, because one of the big concerns that the whole field had was, well, if branaplam is doing this, then is this going to be a risk for all of these Huntington lowering splicing modulators? Are we going to have to abandon the whole idea of a Huntington lowering pill? This slide says, no, we can afford to carry on. We need to keep an eye on it, keep measuring it, keep looking at safety in these patients. But so far, there's no sign of the same risk that branaplam caused. So that's really good news. I will make a very brief mention of Annexon. Annexon is a newcomer to HD, is trying something a little bit different, which is an antibody which is infused into the vein and will hopefully help the health of the synapses, which are the connections between neurons. So it doesn't lower Huntington, but what it might do is help the connections in the brain to last longer. Annexon had some encouraging results from its very small, non-blinded, non-placebo trial earlier on, and is now planning its future trial, which is hopefully going to be starting next year. And I think, one of the really good pieces of news in HD is, if you just look at this wall of names, big pharma, small pharma, biotechs. When I first started in the field, this picture would have had maybe one or two small companies involved. These companies are spending a ton of money on Huntington's disease, and they're doing it because, first of all, they think it's a treatable disease if they can come up with the right drug. Second, they know that this is a community which can deliver. We can deliver on observational research and we can deliver on clinical trials. So, and time again, we've showed that the HD community will recruit trials, run them, and deliver the answers that we need. So I will conclude the early evening news by Zoom with a heartfelt piece to camera. The fact that we're here on Zoom is a slightly painful reminder of the period of history that we've all lived through. The Huntington's disease community was already facing one existential threat. And then we had to deal with the COVID apocalypse. And then it was Ukraine, and then the Middle East, and political upheaval everywhere. It's been a tough few years. And then on top of that, when all of this stuff was happening, we got really disappointing news about multiple drug trials for which there'd been a lot of hope. I want to encourage everyone listening to remind ourselves of a few basic principles. The first is that science is cumulative. It builds on what it has achieved to try to move forward in the right direction. And as long as we keep doing science and doing good science and running good clinical trials, we will always be moving in the right direction, even if we get news that we wouldn't like. As long as we learn from that news, it will help us to continue in our quest and in our shared goal. So 1993, the cause of Huntington's was discovered. That's never going to change. That is the cause. That tells us what problem we need to solve. And that's a huge advantage. The other thing is, we've got all of these companies interested, and that isn't the case. So the pace of research is so much faster. We have so many more shots on goal than we had. And then the other huge thing is the strength and resilience of the HD community. Enroll HD, is it 25 or 30,000 patients now? It's a huge study, and that is a real incentive for companies to get involved in HD. So, and it gives us the data that we need to help recruit trials as well as running them. So if you get the chance, please do take part in Enroll HD. No talk from Ed would be complete without mentioning my own study, HD Clarity, which is a multi-site collection involving collection of spinal fluid to study Huntington's disease and to pave the way for future trials, running at multiple sites in the UK as well as elsewhere. So, and it's built on the Enroll HD platform. Essentially, there will be a day, I believe, where Huntington's disease is understood to be a treatable or even reversible condition. And the purpose of research and of patient and family involvement in research is to bring that day forward as soon as it can be, so that no one has to wait a day longer than is strictly necessary. And a big part of that is patients and families signing up for research. You can always keep an eye on what's happening in the research sphere through HDBuzz, and a good news day is coming. Thank you very much for your attention.